Hello students, in this part we will discuss on different types of DNA and base pair rule. Types of DNA, before we discuss types of DNA, there are some rules on which we can generate we can see the ratio of purine and pyrimidine, means nitrogen base in the DNA. The first one is base pair rule. What is base pair rule? We know that in DNA, purine paired with pyrimidine. Purine paired with pyrimidine. In DNA, purine is two types, adenine and guanine, A and G. And pyrimidine, basically pyrimidine is three types, cytosine, thymine and uracil. But in DNA, there is two types of pyrimidine, thymine and cytosine. In place of in RNA, in place of thymine, it replaced by uracil. But it is the structure of DNA. The base pair rule in DNA is the purine paired with pyrimidine. Purine A and G and pyrimidine T and C. According to base pair rule, purine A bonded with two hydrogen bond with thymine. A adenine bonded with thymine with two hydrogen bond. This is not the sign of is equal to. This is sign for two hydrogen bond. Two hydrogen bond. And guanine bonded with cytosine with three hydrogen bond. Three hydrogen bond. This is the base pair rule. Purine paired with pyrimidine. Purine A adenine bonded with thymine or pyrimidine with two hydrogen bond, and purine guanine bonded with cytosine or pyrimidine with three hydrogen bond. This is the base pair rule. Suppose this is the double stranded DNA. Double stranded DNA means the first strand which has a polarity 3' to 5' and the second one is 5' to 3'. We have already discussed in previous part. DNA, double stranded DNA, both the strands are anti-parallel to each other. Polarity of the both strands are anti-parallel. If this one contains A, T, C, G, then the 5' to 3' A bonded with thymine, T with adenine, C with G and G with C. Adenine with the thymine, two hydrogen bond. This is the two hydrogen bond. Thymine, adenine, two hydrogen bond. Guanine, cytosine, three hydrogen bond, and guanine, cytosine again, three hydrogen bond. This is the base pair rule. One more type of rule is Chargaff's rule. Chargaff's rule. Chargaff's rules applicable only for dsDNA, double stranded DNA. This rule is applicable only for dsDNA. dsDNA means double stranded DNA. According to Chargaff, as per base pair rule, according to Chargaff, in a DNA strand which is double stranded, adenine, the value of adenine is equal to the value of thymine. This here is the not two hydrogen bond. This sign is here is equal to. It means according to Chargaff, adenine is equal to thymine, or you can say that purine is equal to pyrimidine. This is the first one. And second one is guanine is equal to cytosine. here are also there is no symbol of a 3 hydrogen bond. It is stand for is equal. Means the value of adenine is equal to thymine and guanine is equal to cytosine. This is the according Chargaff's rule. The one more rule we can see that according to Chargaff, purine is equal to pyrimidine. Purine is equal to pyrimidine. How? This is the rule applicable for double dsDNA. Suppose this is the double stranded DNA. This one is the double stranded DNA. The first strand which has polarity 3' to 5' and second one is 5' to 3'. Suppose here is A, T, C, G and C, or you can say that G again, or ATCG. Then the opposition end, A bonded with thymine, P with A, C with G and G with C. According to first one, A is equal to T, means adenine is equal to thymine. The number of adenine and thymine we can calculate. Adenine is 1 and 2. 2 adenine. A is equal to 2. And thymine is 1T and 2T. So T thymine is equal to 2. So according to Chargaff, A is equal to T, or you can say that A by T value is equal to 0. 1. That is A की value कितनी 2, T 2, 2 by 2 is equal to 1. This is the proof कि adenine is equal to thymine. And second, G is equal to C. Again we here add C, then here is G. Count the number of guanine and cytosine. This is also G is equal to C. It means G by C, the value is equal to 1. Calculate the value of C. 1, 2 and 3. There are 3 C. And G 1, 2 and 3. G is equal to 3. So we can say that C by G is equal to 3 by 3. Yeah, G by C is equal to 3 by 3, that is equal to 1. So according to Chargaff, purine is equal to pyrimidine. Clear? To show the value of purine and pyrimidine is equal to, he put an example. Take an example of a DNA. DNA, which one? dsDNA, double stranded DNA. Suppose a segments of double stranded DNA, it contains adenine is 20%. What are the rest of the components in the dsDNA segments? Suppose in a dsDNA segments, the value of adenine, nitrogen base is adenine 20%. What are the components? What are the value of the rest components in the dsDNA? We know that adenine is equal to T according to Chargaff rule. If A is equal to 20%, then T is also 20%. The total value is 40%. The rest value remains. The rest value of DNA is the 100% and 40%, 20% adenine, 20% thymine. The rest value is 60%. And purine is equal to pyrimidine. We know that A is equal to T, Z is equal to C. So we can say that G and G and C both value कितनी 60%. Both are equal. Both are equal. It means G is equal to 60 by 2, 30%. If G is 30%, then C is also 30%. We can calculate the value of the nitrogen base pair if one is given. This is the two base pair. One is base pair rule and the second one is the Chargaff. In next we will discuss types of DNA. Types of DNA. DNA is different types on the basis of different techniques. First one is on the basis of function. On the basis of function, the DNA is two types. This is the first types of DNA means how is the DNA is different types. First one is the basis on the basis of function. The DNA is of two types. First one is trophic DNA and second one is genetic DNA. Trophic DNA and second one is genetic DNA. What is the major difference between trophic DNA and genetic DNA? Trophic DNA, the DNA which is applicable, which is responsible for all the activities in the body, such like as digestion, respiration, excretion, but not involvement in reproduction. The DNA which is responsible for all the activities in body but not for reproduction. The DNA which take part in all the activities of the body but not in reproduction, that type of DNA is called trophic DNA. And genetic DNA means the DNA which is responsible, which take part only for reproduction, means inheritance. Genetic means inheritance. The DNA which is responsible in reproduction not for other activities, that DNA is called genetic DNA. We can calculate. So this type of DNA, genetic or trophic DNA, is applicable not in higher organism, it is applicable for the lower categories organisms, such like an example of paramecium. For example, paramecium. In paramecium, this is the structure of paramecium. It contains cilia outside the body. In paramecium, there are two types of nucleus, or there are two types of nuclei. One is large, that is called macronuclei, and other one is small, that is called micronuclei. One is large, that is bean shaped, and just near to the bean shaped large nuclei, one is another nuclei that is spherical, small in size. This large nuclei is known as macronuclei, and this smaller is known as micronuclei. As the paramecium division in the paramecium can take place, this macronucleus disappear. It means it dissipates into the cytoplasm of the paramecium. It disappears. It doesn't take part in the reproduction. But as the division in paramecium can take place, the small nuclei divided into two parts, and that goes into the two organisms. So this small nuclei take part in reproduction, which is known as a micronuclei, and micronuclei act as a genetic DNA. Whereas macronuclei, when the division in paramecium can take place, this macronuclei disappear. So it doesn't take part in reproduction. This is called trophic DNA. Again listen, the trophic and genetic DNA is applicable. This one is trophic DNA is applicable only for lower categorized organism, not in higher organisms. This is the first base on the basis of which we can categorize the different types of DNA. The next one is on the basis of base pair and coiling structure. The types of DNA is 5 types. On the basis of base pair and coiling structure. The DNA is 5 types. Which one? DNA A, DNA B, DNA C, DNA D, and DNA Z. These are the 5 types of DNA on the basis of base pair as well as coiling structure. How they are coiled, clockwise or anti-clockwise. First one is base pair. DNA A, which we have, DNA A, it contains only 11 base pair in one turn. In one turn, DNA A contains 11 base pair. In previous part, we have discussed about J.D. Watson and F.F. Citric double helix structure. In that one turn, that contains 10 base pair. That was the DNA B. It contains 10 base pair. The mostly common DNA is DNA B. DNA C, it contains 9.33 base pair. How this 33? One turn contain 11, that all the whole segments of nitrogen base pair present. 10. How this 9.33? It means when the one turns from when we start in a helical or a spiral stair from ground floor to upper floor at the 360 angles, there are some parts of nitrogen base may be suspended into the one turn, so it is counted as 9.33. DNA D, it contains 8 base pair, and the DNA Z, that contains 12 base pair. So on the basis of base pair, there are 5 types of DNA, such like as coiling or rotation method or rotation direction. DNA B is right-handed, which we have discussed Watson and Crick model. So DNA A right-handed, you can say that clockwise. It is twisted clockwise or right-handed. DNA B is also right-handed, clockwise. DNA A right-handed, clockwise. DNA D, that is also RH right-handed, clockwise. But there is dissimilarity in DNA Z. DNA Z is left-handed anti-clockwise coil. So mostly questions are asked from 2 types of DNA Z, because it is dissimilar out of the other 4 DNA. And the other one is DNA B, which is common in all DNA. So the questions are asked on the basis of DNA B and DNA Z. This is the different types of DNA on the basis of function as well as base pair or coiling structure. Next to process, some terms are also which are useful for us in this chapter as well as in biotechnology. That is, first one is palindrome. What is palindrome? Palindrome is a word or phrases of a word. Palindrome is a phrase or word in which we have to read. We have to read backwardly or forwardly. The sequence remains same. It is a phrase which when we read whether forwardly or backwardly, the sequence remains same. The sequence remains same. It means when we read out the double DNA, dsDNA, nitrogen base pair, the sequence of base pair remains same in the same direction. Same direction means if we read out one code from 3' to 5', then we have read the same code from the same direction 3' to 5'. Suppose this is the dsDNA and it is the sequence 3' to 5'. Sequence contain AA, ET, CG, AA, TT. Oh, sorry, TT, AA. Or you can say that AATT कर दीजिए, AATT. And this is the another. This is the 5 prime. Sorry, 3 prime. The another one is 5 prime. This is the 5 prime. This one is 3 prime. As we read this code from 3 prime to 5 prime, AATT, CG, AATT. Then we read from same sequence 3 prime to 5 prime. Means this is the forward link and this one is the backward link. Then we know that T bonded with A and A with T, G with C, C with G, A, फिर again, then T. If the 3 prime of first strand sequence is AATT, CG, AATT, then the sequence of 3 prime is also 3 prime to 5 prime, AATT, CG, and AATT. You can also take an example of Malayalam, Malayala. If we read this sequence from here to this one, Malayalam, then we read here from this one, this also contains Malayalam. The sequence of phrase, or you can say the phrase of words, when we read forwardly or backwardly direction, the sequence remains same. That is known as a palindrome. This is very helpful in the biotechnology chapter when we read out. The second one is DNA has the property of denaturation and renaturation. Denaturation and renaturation. Not denaturation, degeneration भी कह सकते हैं. Degeneration and regeneration. At the suitable temperature, dsDNA, it remains the same, 3 prime to 5 prime and 5 prime to 3 prime, attached with a nucleotide. Means the backbone attaches both each other. What is backbone? This strand, both the strands which are made up of pentose, sugar and phosphate, they don't contain nitrogen base. Pentose, sugar and phosphate. These are the backbone. When we hit this temperature, when we hit the DNA at the exceed temperature, normal temperature से ज़्यादा यदि DNA को हम heat out कर देते हैं, means heating process करते हैं, और heating process करते हैं, then both the strand of DNA is separated. Then both strand of DNA separated. This is the 3 prime, 5 prime and 5 prime to 3 prime. This is called denaturation. When we heated the DNA piece at exceed temperature than the normal, then both the strands of DNA separate from each other. This is called degeneration or also denaturation. This process will be taken in biotechnology when we do the process of PCR, polymerase chain reaction. This we will discuss in biotechnology. And again, when we provide a suitable temperature, when we provide this both the strands suitable temperature, both the strands also combined, both the strands backbones combined, and this is called regeneration or renaturation. So this is the property of DNA. This is one, the first one palindromic, and second one is denaturation, degeneration or denaturation, regeneration or renaturations. These are the two words, two terms which are discussed when we study about the DNA. Clear? In next part we will discuss on the next one.